Welcome back, grade 12 to Maths and Science series, Life Sciences with Spongile. So previously we had went through the cell, right? We went through the nucleus and we went through how it condenses the chromosome and what we find inside the chromosome when we look through the microscope, which is the DNA structure, which is double-stranded and has a shape of a helix, a double helix. So now I'm going to give you a little bit of history about the discovery of DNA. So it was discovered by James Watson, Francis Crick, using the data that was provided to them by Maurice Wilkins and Rosalind Franklin. So your teacher will probably tell you about this other part about Rosalind Franklin. So she was a scientist, female scientist, who was very much facing patriotism in the highest. So she's actually the one who discovered DNA. And then she went to her male colleagues, which were Watson and Crick and Wilkins, and they decided to take credit for her work. And somehow it was never corrected. So every time in an exam, they ask you who discovered, they want you to give the answer Watson and Crick. And do not mention Wilkins and Franklin. Okay? Hope you get that, hey? And now we're moving to the structure of DNA, which I told you we are going to do, by the way, right? So look closely there. I need you to look as closely as you can because this right there is magic. Sorry there. Look at that, the structure of DNA. This is how they're going to give it to you in the past question papers. You see that little structure there? It will just carry on elongating and elongating on the question papers. And so, like I said before, the structure of our DNA molecule is double-stranded, but the shape is double helix. Now, I made a little key there because I want us to know a little bit of a small structure that is called the nucleotide. So the nucleotide is this little structure here all together like that is what we call our nucleotide. So this small circle here is called the phosphate group. And then the pentagon that you see here, since we are on the DNA, we call it the deoxyribose sugar. But if we are not on any of the nucleic acids, we call that pentose sugar. Okay? And then those red dotted lines between the nitrogenous bases is called weak hydrogen bonds. Remember, when you first saw me talking about this topic, I did give you a terminology, and among them was weak hydrogen bonds, where I was telling you that these are the bonds found between nitrogenous bases. And now I need to tell you that these are the DNA nitrogenous bases. So we have thymine, guanine, adenine, and cytosine. And something unique about these nitrogenous bases is that they complement one another. So what am I saying to you? I'm saying to you that adenine will always, adenine will always go with T, and then G, guanine, will always go with cytosine. And this is in DNA. This part only happens in DNA. When we get to RNA, this changes, okay? We only change this part and we put a U there. But this stays the same throughout both of the nucleic acids. So the nitrogenous bases are complementary in the sense that adenine goes with thymine and guanine goes with cytosine when they pair. I did that pairing here. So thymine goes with adenine, guanine goes with cytosine, and they are bonded together by the weak hydrogen bonds. I hope we're together there because the following is the structure of RNA, which I told you to be ribonucleic acid. So its shape, it is single-stranded. You understand? It is single-stranded. As you can see, it only has this part only, whereby this one was double-stranded. It had this one strand and this second strand. and this one only has one strand. And I said to you, we call this little structure here all together like that a nucleotide. I told you what is a nucleotide previously. I said to you it is a monomer of both the nucleic acids. As you can see, the structure is here and it is also present here. You see that? So now we have similarities. They both have the phosphate groups. And which are those circles there? That circle there. They are phosphate groups. And then now, look at the change. Here we said it is deoxyribose sugar, right? That pentagon sugar there. It is called deoxyribose sugar in DNA. But when we get to RNA, it is called ribose sugar. And then I told you that there's going to be a certain change as well in the nitrogenous bases. You see there, we now have uracil instead of thymine. Exactly. Now that it means that here, adenine will pair with uracil. And then guanine stays with pairing with cytosine. You see? Don't forget these. These are very important. You need to note these changes down because they're examinable. They will ask you to tabulate the differences between DNA and RNA. And that is a few that I've already given you. I told you that firstly, DNA is double-stranded, and then it has the deoxyribose sugar, and then it has weak hydrogen bonds. As you can see, those bonds are not present here because RNA is single-stranded and has ribose sugar, and its nitrogenous bases are not pairing with each other though. You see that? There's no presence of weak hydrogen bonds. And that right there was it for the structure and the history of DNA and the structure of RNA. Bye. Thank you very much. Don't forget to like and subscribe and comment and keep resharing to keep motivating us and not keeping this knowledge to yourself so many other kids can get it. Toodles!